Hello and Assalamualaikum everyone. You are with me, Madam Waishas, and in this video, we are going to learn about chromosomal mutation. First, learning outcomes. We are going to define chromosomal mutation and also state two types of chromosomal mutation, which are changes in chromosomal structure or also known as chromosomal aberration, and also changes in chromosomal number. Definition: abnormalities in chromosomal structure and changes in chromosomal number. It can be passed to offspring if mutation occur in gamete cell. It also can increase variation among individuals. It may occur when chromosomes are being pulled apart in mitosis or meiosis, crossing over in prophase 1, and also DNA replication during interphase. Types of chromosomal mutation: we have chromosomal aberration and also chromosomal number alteration. Let's have a look at chromosomal aberration. It means rearrangement of a certain segment or parts of chromosome are most frequently formed during mitosis or meiosis. We have inversion. An inversion reverses a segment within a chromosome. Next, deletion. A deletion removes a chromosomal segment. Duplication. A duplication repeats a segment. And the last one, we have translocation. A translocation moves a segment from one chromosome to a non-homologous chromosome. Alright, so next part we are going to have a look at chromosomal number alteration. Learning outcomes: state the types of alteration. We have Aneuploidy and also Euploidy, and also state the Aneuploidy effect on autosomal chromosome, monosomy 21 and trisomy 21, and also sex chromosome. We have Klinefelter syndrome and Turner syndrome. First definition: changes in chromosome number due to extra or missing one or a set of chromosomes. Types of chromosomal number alteration: we have Aneuploidy that can be divided into monosomy and trisomy, and also we have Euploidy or Polyploidy that can be divided into allopolyploidy and allopolyploidy. Aneuploidy, based on Campbell, a chromosomal aberration in which one or more chromosomes are present in extra copies or are deficient in number. While based on Solomon, any chromosomal aberration in which there are either extra or missing copies of certain chromosomes. Causes of aneuploidy: both set of chromosome pass to the same pole of the cell, or non-disjunction in anaphase 1 or anaphase 2 during meiosis. Sister chromatids fail to separate and move to the opposite poles during anaphase 2 of meiosis 2. Or maybe failure of pair of homologous chromosome to separate and to move to the opposite poles during anaphase 1 of meiosis 1. Normal human cells have 46 chromosomes, 22 pairs of autosomes, and 2 sex chromosomes. This is called the diploid number 2n. Female carry 2 X chromosome while male have an X and a Y chromosome. Autosomal abnormalities, monosomy 21. Characteristics: short distance between eyes, large ears, and contracted muscle. Autosomal abnormalities, trisomy 21. Characteristics: short stature, broad face and slanted eyes, weak muscles, flattened nose, large protruding furrowed tongue, small irregular teeth, suffer mild to severe mental retardation. Next is sex chromosome abnormalities, which is Klinefelter. Characteristics: sterile male with small testes and fail to produce sperm, feminized voice, breast enlargement, long hands and legs, and they have normal IQ. Next is Turner syndrome. Characteristics: sterile female whereby they fail to ovulate, small breast, dwarf, short stature, and webbing of skin of the neck. Alright, next we are going to learn about chromosomal number alteration, Euploidy. Have a look at the learning outcomes. Okay, we are going to state the type of alteration, Euploidy, or also known as Polyploidy. First, definition. Changes of the chromosomal number which involve the whole chromosome set. It occurs when the whole set of chromosomes did not separate during gametogenesis. It is common in plants than in animals, and the gamete fusion will produce cell or organism which has more than two sets of chromosomes. Alright, next, types of polyploidy. We have autopolyploidy and also allopolyploidy. But first, cell with only one set of chromosome is known as monoploid, but it usually rare and cannot survive. At the same time, cells with 3 or more chromosome sets are called polyploid cell. Let's have a look at autopolyploidy. An individual that has more than 2 chromosome sets that are all derived from a single species. It can caused by non-disjunction in meiosis that cause the formation of diploid gametes. The fertilization of a normal gamete haploid with diploid gamete will produce triploid organisms. Autopolyploid plants produce flowers and fruits bigger than normal diploid. Next is allopolyploidy. A fertile individual that has more than 2 chromosome sets as a result of 2 different species interbreeding and combining their chromosomes. F1 hybrids are usually sterile because haploid set of chromosome from one species cannot pair with haploid set of chromosome from the other species. Chromosome number in sterile hybrids becomes double and produce fertile hybrids. Allopolyploid will produce new species. Alright, that's all. Thank you. Bye-bye.